Bonjour tout le monde. Merci encore d'être venu pour notre session de séminaire à l'IBIS. Aujourd'hui, on a l'honneur d'accueillir Dr. Sarah Carey. Elle est présentement stagiaire postdoctorale de United States Department of Agriculture National Institute of Food and Agriculture, USDA NIFA, à Hudson Alpha Institute for Biotechnology in Huntsville, aux États-Unis. Elle a fait ses études en biologie et obtenu un doctorat en botanie de l'Université de Floride et depuis travaille sur les questions quand même intéressantes de séquençage de génome et spécifiquement d'identification des chromosomes de sexe qui sont parfois très complexes chez les plantes. On est bien content de l'avoir ici et je vous laisse pour la présentation. Thank you, Sarah, again for accepting our invitation and be here. And stage is yours. Thank you.
Awesome. Thank you so much. Thank you all for attending and for the offer to talk with you about some of the research that I've been working on. Can you hear me okay? I didn't mute. Awesome. Okay. So yeah, I'm going to talk today about some of the exciting projects we've been working on across plant sex chromosomes. First thing I want to do though is take time to thank a lot of my really awesome collaborators. So I work with Dr. Alex Harkis. I'm a postdoc in his lab at Hudson Alpha, and we have an incredible team of folks that I have the opportunity to work with in Alex's lab. Our lab manager, Haley Hale, and Hannah Mueller, our lab technician. They do almost all of the molecular work that I'll be talking about today, and without them and their skills at extracting DNA, giving us really good quality DNA, it would make my job as a genome assembler much more complicated. And then we have awesome grad students in our lab, Zach Maharg and Alermia Kozbek. They both work on dioecious projects with me. And then the folks in Jane and Jeremy's group, like John Lovell and Jerry Jenkins, they're really great mentors on some of the analyses that I'm going to talk about. Les Goertzen is an incredible colleague at Auburn who has helped field collect with a lot of the plants that I'm going to talk about a lot of those ones today, but it's still this is like larger dioecious project he's been instrumental in. And I'm going to talk a little bit about hops, and that's much thanks to like Josh Hevel and Gary Muehlbauer and Catherine Easterling, and also thanks to Jim Liebens-Mack, who is the lead PI of the Open Green Genomes project that I'll talk a little bit about, and my PhD advisor, Stuart McDaniel, because I'll be talking about some of the moss projects we've been working on today. So just to start out, I want to tell you about what dioecious is. This is absolutely my favorite trait in plants. It's very cool. It's where carpels and stamens develop on separate individuals. And right now we're focusing on flowers. And so this is really rare in flowering plants. Most flowers are perfect, or maybe monoecious, but most are perfect, where you have whorls of sepals, of petals, of carpels and stamens all in that same flower. With dioecious, males and females of the species will have both all the petals and the sepals, but only females will develop functional carpels and only males will develop functional stamens. And so that's what dioecious is, when you have separate sexes on separate individuals in plants. And so you've probably encountered some dioecious plants in your life. Your refrigerator is probably full of some dioecious plants. If you like soda pops, like root beer and sarsaparilla, some of those main ingredients that give those wonderful flavors are actually dioecious, like sassafras and smilax. If you enjoy beer, the hops that gives the bittering agent is also a dioecious plant. And some of the fruits and vegetables and nuts that you might enjoy are also dioecious, like spinach, kiwi fruit, asparagus, and mulberries. Your spice cabinet is also full of dioecious plants. So black pepper, nutmeg, allspice, and bay, these are all dioecious. And so you can kind of get an idea that there's like a really wide breadth of diversity in species that have this. They are peppered across the phylogeny, these evolutions. And so kind of a big question that we're hoping to address as much as we can is truly how many evolutions of diaceae are there across plants? This is going to be a long-term question because 5 to 10% of species in flowering plants have this trait, and there's 400,000 angiosperms, so it's quite a few to test every one, but it's one we want to tackle as much as we can. And I'll be talking about some of these evolutions in the talk today. And then the other thing that we're really interested in Alex's lab and that I'm working on is what genes are associated with this transition from having a perfect flower to being dioecious. What genes cause carpel and stamen sterilities at some points in the pathway? And there's been some amazing work that has been done and functionally verified in several species to uncover some of these genes. So in things like kiwi fruit and asparagus and grapes, been able to identify some of the genes that are involved with these carpel and stamen sterilities. Kind of the most important thing I want to point out about this particular figure, though, is so far every gene that has been identified is different. There's not the shared evolution of any one gene that we've found yet. And a lot of times so far, things hit different parts of the pathway. So in asparagus, for example, the diaceae genes, in one of them interacts or affects tapetum development, while other ones, like in grape, affect pollen production. So it's different parts that kind of have been evolving this role across flowering plants. And so something we're really curious about is what other genes are involved with other evolutions that hit other different parts of the pathway. It's another kind of broad long-term question, but one we'll be talking about today. And so because diaceae is involved with the evolution of separate sexes, there's very often sex chromosomes that are underlying this evolution or that will evolve after diaceae evolves. Not always, but often. And that kind of gives us a nice area of a genome to narrow in on to find these diaceae sterility, carpel and stamen genes. And so when we think about what sex chromosome, what we're looking for is that non-recombining region that has those sex-determining genes that's only inherited through, in this case, an XY, so only inherited in males. And because I do genome assembly to first be able to identify these diaceae-related genes, the goal for a lot of our projects is to be able to have completely phased assemblies, to have both the X chromosome and that Y chromosome completely phased out and contiguous. And so what we want is a representation of the genome that's either a haploid assembly of the autosomes and both sex chromosomes are available. This is kind of how the human genome has been represented over time, if you're familiar with that. Or what things are moving to more now with the updates in technology and sequencing and assembly side of things is having a full diploid representation of a genome. So having all of the autosomes represented completely, as well as the X and Y chromosomes. But for the focus of sex chromosomes, just really want to be able to have both the X and the Y completely cleanly phased out. This is very complicated to do, though. Sex chromosomes are notoriously complicated to assemble. And one you may be familiar with is with the human XY system. The telomere to telomere, which is the complete human genome assembly, was released earlier this year. But that first initial publication, the Y chromosome wasn't there. It took a few more months for that to be released. It was the last chromosome to be released in the assembly. And even though it's a very small chromosome, it's because it's very, very complicated. So when you look at the autosomes, there's tons of gene content. There's maybe some heterochromatin, and there's plenty of repeats for sure. But when you look at the Y chromosome compared to the X, there's a huge non-recombining region. It's 95% of the chromosome. Not much gene content. It's mostly ampliconic, mostly heterochromatin. And those are all things that make it very, very complicated to assemble, especially the repetitive nature of them. But to add another level of complication when working on plants, not all sex chromosomes are like the human XY. If they were, it would be hard, but we at least kind of know what we were aiming for. Across all of the other systems that have been studied, the sex chromosomes vary wildly. So for example, that size of that non-recombining region can vary wildly across them. So asparagus is an example of one. is a very, very, very small non-recombining region. It's about 1% of the chromosome that it's on. And it kind of, there are some that are a little bit bigger. So like willow, it's around 45% of the chromosome. See, humans here are up at that 95%. And one of the moss species that I've worked on, it's nearly the entire chromosome, as far as we could tell, does not recombine. So, the area in which we're trying to identify that non-recombining region can be quite different in size. In addition to that, not all sex chromosomes are XY. Some are, like hops, where inheritance of two X's develops females, and inheritance of the XY develops males. And so that Y chromosome is only ever inherited through males in the species. There are some species of plants and animals too, but in plants where instead of the males being heterogametic sex, it's the females. So males inherit what we call 2Z chromosomes, so they're ZZ, and the females are the ones that have that sex-specific chromosome that's only ever inherited through females. There's another sex chromosome system out there that exists too, and I'll talk a little bit about mosses at the end, but this is just another level of fun complication, is that mosses and other bryophytes, they are haploid when they are expressing sex. So instead of being XY or ZW, they have what's called a UV system, where inheritance of a single U chromosome develops females and a single V develops into males. It makes it a little bit easier when you're thinking about a genome, because you don't need to target the heterogametic sex per se. If you assemble a female, you're getting the U, but still finding those non-recombining regions can be challenging. Another level of complication, just keep adding them on because these are really complicated parts of the genome, is that not all sex chromosomes can even be found by cytology. Hops are one of the earliest examples of ones that were found because they're what's called heteromorphic, where you can see in the karyotype here, these are the 9 autosomes, and then the XY chromosome. That Y chromosome, just like in humans, is much smaller. So it was able to be identified as that chromosome that only ever was inherited through males, and thusly was the sex chromosome pair. But it turns out most plants don't really have that. Most of the sex chromosome pairs are what's called homomorphic. So at least in terms of looking at the karyotype, it's indistinguishable. You can't tell whether which pair is a sex chromosome, and then being able to figure out whether it's an XY or ZW system is not possible without additional analyses. So this is something that we've been working on trying to address, something we've called sex chromosome cytogenetics by sequencing. So trying to uncover the cytotype of a sex chromosome pair using some shotgun data. It's really inspired by the amazing cytologist Nettie Stevens, who's one of the first people to identify sex chromosomes in mealworms. She found the XY pair in a lot of insects. And so in absence of there being actual cytological differences, we were trying to use DNA sequencing as our microscope to find the XY and ZW sex chromosomes. And so there's two goals for this cytogenetics by sequencing we've been working on. One is to identify if a species has XY or ZW sex chromosomes using cheap Illumina shotgun data. That way we can target the heterogametic sex for our genome reference. And then once we have that pseudomolecule scale reference, use those same data in order to find that non-recombining region on that sex chromosome pair. And so what we've been doing with this pipeline is using something called k-mers, which is if you take short read data or any data, you can pick an arbitrary size of DNA. So I typically use 21 mers. And these programs will find every single one of those 21 mers, so 21 base pair bits of sequence in your data. And what I do is I look for whether males or females have more k-mers. And the idea is if it's an XY system, because all the autosomes and the X chromosomes should be shared between males and females, the only genomic difference, if we compare across males and females, should be that non-recombining region in the X. So there should only be k-mers coming from that non-recombining region in an XY system. So if I find more male-specific k-mers, it should indicate an XY system. If instead I find more female-specific k-mers that's coming from that non-recombining region of the W chromosome, and then should be a ZW system. And so I've tested this on quite a few species, sometimes as low as 3 females and 3 males. A few of these, there's 4, just because the data was available. But what this plot's showing is if the positive axis that I found, there's more male-specific gamers that were identified, and so indicate an XY system. And then in the negative direction, there were more female-specific gamers, and so indicate a ZW system. And many of these were already known systems, so testing it in things where we were already sure that we had an XY or ZW, like Morella rubra, the strawberries, and then Amberella. And then the new one that we've been able to identify is another species of Morella, which is a project led by Laramia Kozbek in our lab. And then many XY systems have been found using this. So verifying with things like papaya and cannabis, where we already know there's an XY system. And I've found a few new ones. So in Baccharis halamifolia, there's evidence for an XY, as well as in so Myrothamnus flabellifolia is a new identification. And so already making some pretty good progress on being able to find out which is the heterochromatic sex using shotgun data only. And so the next part of this pipeline is, once we know which is the heterochromatic sex and have a target for our genome assembly and build that out, we want to be able to identify that non-recombining region in the genome. So find the sex chromosome and find that region that is the non-recombining bit so we can find our sex determining genes. And so what we really want is, like I was mentioning before, we want a phased assembly. So autosomes, either a single copy of them or both there, but really, really want that X and Y completely phased out. What happens though when you do a de novo assembly is you get 2 haplotypes out, but you don't know which is the X or the Y, haplotype 1 or haplotype 2. So you have to figure that out using some analyses. The bigger issue though, additionally is oftentimes these regions can still end up chimeric without additional analyses, so that both haplotypes are kind of a mix of the X and the Y chromosomes. The biological parental haplotypes have switched when you go to completely scaffold the assembly. And that's a little bit, takes a little bit more computational work, but it's something that we've been able to fix when we run into it. But so, excuse me. An example of one of the species that we've been working on and have been testing this pipeline on is Amberella trichopoda, which I like to call the platypus of flowering plants. And there's a few reasons it's very similar to platypuses. It's a monotypic species. It's the sister lineage to the rest of flowering plants, so it has a really important phylogenetic position. And then it also has relatively weird sex chromosomes. And so this is a genome that's part of the Open Green Genomes project through the Joint Genome Institute, and was done by Jerry Jenkins here at Hudson Alpha and his team. And so Jerry's been able to assemble, using a combination of PacBio Hi-Fi and Hi-C data, all 13 of the Amberella chromosomes. And so we have 2 very well-phased haplotype assemblies. There were very few contigs at the beginning, so these assemblies are getting really much easier to get together. Really big pieces that we only have to kind of put a few together in order to get whole chromosome arms if the whole thing wasn't completely assembled at the initial assembly. And the assembly lengths are about the same. So we have pretty much full representation or close in both haplotypes. And so my task after the assembly was to figure out which haplotype has the Z or the W. Where is that non-recombining region, and is it fully phased, or did it end up chimeric? And if it's chimeric, how do we fix it? And so this is where those sex-specific k-mers come in again. So I found all of the female-specific k-mers because this is a ZW system. And I used several different population types because I was really rigorously trying to figure out what we need in order to do this. So there's a few that are within populations. So I have some within, so Amberella is all from New Caledonia. So these are different populations on New Caledonia of Alpini and Dogny. I use an island-wide sampling, so males and females from across the island, kind of just whatever existed, as well as a mapping population. So in theory, there should only be a single W chromosome in the offspring. And then I tested if we do 3 males versus 3 females, 5 males versus 5 females, or just all of data we have available, just to see, is 3 not enough to be able to find these regions well, and is 16 too many? Do we lose too much because it's low coverage data? And it turns out with Amberella that any of the analyses I find point to this is on chromosome 9, and there's a very strong conserved region, or a core region that I can find using any of these comparisons here. So it shows that haplotype 1 on chromosome 9, right around here, on the 44 megabase region is that sex chromosome. And if you look over at haplotype 2, you can see a few peaks of things. There's a little bit of noise, but that's not totally unexpected when you're mapping things that are 21 base pairs, but there might be a few places that go, but very clearly it's not a nice peak. It's noise at best. And so haplotype 1 is that W chromosome and looks pretty well phased. The reason why I really like k-mers is because some of the other analyses that exist are great for finding the sex chromosomes, but it's not as easy necessarily to tell which is the Z or the W when you have both available. So when you think about read coverage, which is kind of one of the classic ways to do this, especially in animal systems, there's a region here you can kind of see an example of this is clearly W specific right here, because males don't have any coverage in that spot. Females, since they're Z and W, have half coverage in that spot. But most of the rest of this non-recombining region, the reads map equally well between the Z and the W, so you can't use coverage. And you can see that pattern holds for both haplotypes. So being able to figure out which was the Z and the W and was it fully and contiguously assembled is coverage won't be able to do it. The complementary analysis to coverage, so if your reads map is to look at nucleotide diversity between the sexes, and you can see that also has a similar pattern between haplotypes 1 and 2. So being able to tell if this was a chimeric assembly would still be much more challenging using nucleotide diversity. And instead, the k-mers make it very, very clear that this is the W chromosome and it's not broken up and it's not chimeric. And just to show you an example of what I mean when something is chimeric, this is our phased assembly, and that's just the mapping population data shown now. But when you map to a single haplotype version of the assembly, so this was the previous build, and instead of phasing out both chromosomes, it was a single haplotype representation where you expect that maternal and paternal haplotypes will be switching in and out of what's ultimately assembled. You can see that it is truly chimeric. So there's a bunch of W-specific data where all of these peaks are, but it drops off, and that region there is going to be Z, and then you're back to W, back to Z. And so that's what chimeric looks like, and that's often what we've been running into when you assemble sex chromosomes in a single haploid into a single haplotype. But we are able to assemble them into phased references now and find them. The nice thing about AMBO was this fell out in one contig, so it made it really cleanly assembled. There are a few things we're working on that region is maybe 3 contigs, and it gets a little more tricky, but I think we have the tools in order to be able to pull those together. And so now that we have this amazing reference, the sex chromosomes are phased in Amberella, you get to ask the fun questions of when did they evolve and what are those sex determining genes? And we already kind of knew how old the Amberella sex chromosomes were. This is previous analyses that were done. And so Amberella is, like I mentioned, the sister species to the rest of flowering plants. It diverged about 130 million years ago, but based on kind of KS analyses or protein evolution analyses, the sex chromosomes evolved 16 million years ago. So relatively recent compared to things like mammals. And so the next thing we're working on is what are the sex determining genes? There are about 100 that are annotated in that non-recombining region. And so I'm doing a bunch of fun analyses to try and narrow in on those potential candidates. So in addition to Amberella, I've been starting to work, this is what my postdoc USDA project is on, and on the hops sex chromosomes. And so hops are really cool for a lot of reasons. One of them being, like I'd mentioned earlier, they're one of the earliest plants that had sex chromosomes identified, and that's because they have a heteromorphic X chromosome or Y chromosome in males. But that's only one variety that has that heteromorphy. It turns out that some of the other varieties are instead homomorphic. So they're the same size. And so it's curious, like what happened to cause either gene loss in one of those varieties, or is there an interplay between gene loss and TEs and one of the other ones? It's gonna be a really fun molecular kind of comparison study between those. But there's also multiple systems in other varieties. So some of the varieties of Humulus lupulus have a double system where there's 2 X chromosomes and 2 Y chromosomes. And then there's another, there's actually the sister species, Humulus japonicus, that has an X and 2 Y chromosomes. So just a lot of really cool cytological differences that lend themselves to some very fun molecular evolutionary questions. But unfortunately, I don't have the data back in order to talk too much about them. Sadly, I was hoping I would, but we're working on assembling those so that we can figure out the sex-determining genes and get to all this fun analysis. Excuse me, all the fun analyses. In the meantime, though, there are other references that exist in the Cannabaceae family and as well as the Moraceae family. So kind of getting back to that question of when did they evolve and how many times have sex chromosomes evolved, leveraging those existing genomes to start tackling some of that question. And so hops and hemp, they share an XY sex chromosome pair that's was discovered in this printout paper. And so using cannabis as kind of the reference for what the hops genome would be, wanted to ask, does cannabis have the same sex chromosome as any of these other dioecious species in the Moraceae family? So there's mulberry genomes, there are fig genomes, and working on an Osage orange genome to add to this as well. All of them that have been studied all have XY systems. There's been some ancestral state reconstructions that suggested that it's a shared, that dioecious evolved in this ancestor to these two families. So are they the same sex chromosome? And so what I've done is I've used existing genome assemblies and done some synteny analyses. And what this is showing is these are blocks of genes that are in the same syntenic order. So it indicates homologous regions of chromosomes based on gene order. And I've put an X where the sex chromosomes are. And so based on existing analyses from papers. And so if we look at Cannabis sativa, this is the CS10 reference. This chromosome here is the X. And you can see that it's syntenic with this one in the mulberries and this one, these four actually in the figs, but those aren't the sex chromosomes. So pretty clearly, this is not the same homologous chromosome with the two species in the Moraceae family. And what's also really interesting is when you look at these two species in Moraceae, the non-recombining region is in different parts as well. So this is the same chromosome in mulberries as this one in fig, but the actual non-recombining region is in a different part. So is it a different chromosome? Did it translocate? That's the same question for cannabis. Was it a single origin and this is a translocation event? It's the same sex-determining genes that have kind of been moving around, or is it completely different evolutions? And that's kind of the next question that I'm going to start tackling across these two families. But cool nonetheless that they appear to be different chromosomes. Okay, so those are the really fun projects we've been working on, currently assembling genomes on flowering plants. I'm going to switch gears and talk about some moss sex chromosomes now. And mosses are just such a cool group of plants, and I wish I got to work on them more still, but maybe soon. There's a lot of things that are different about mosses than the flowering plants. One of them being the proportion of dioecious is much greater. So 60% of mosses are dioecious. Technically, it's called dioicous because they're haploid, but just for simplicity, going to use dioecious through here. And in mosses, instead of making flowers, they make structures called archegonia in the females and antheridia in the males. So the archegonia are the egg-producing structures, and the antheridia are the sperm-producing structures. And those are on separate individuals. And ceratodon is very cool because it has a lot of different sexual dimorphisms. And you can see this here in this picture, where the female on the left it grows much larger than the male. So it's a really neat system. But of course, my favorite thing about ceratodon is it has giant sex chromosomes. They are 1/3 of the genome in the species. And so here's a squash of ceratodon. There's 13 chromosomes. And the one I have indicated here is the sex chromosome. And so you can imagine that a giant sex chromosome probably was really hard to put together into a genome. And so this was another JGI project that I've been a part of. This is work that I did in my PhD lab with Stuart McDaniel. And the Ceratodon genome started when Stuart was a postdoc. This was a 12-year-long genome project because those sex chromosomes were so complicated to put together. And it makes sense. The genome's small, it's only 360 megabases, but when 1/3 of the genome isn't recombining, all of the tools that we previously had in order to put things back together weren't going to work. But once PacBio and Hi-C data started coming around, this is another assembly by Jerry Jenkins and his team from Jeremy's lab. They were able to pull it together and assemble us both a male and a female reference. So we have both the V and the U chromosomes assembled for this species. Yeah, there it is, sex chromosome. And why are these chromosomes so big? There's a couple reasons. One is they do actually have a lot of genes on them. So we were able to annotate over 3,400 genes on each chromosome. So both the U and the V had about 3,400 genes. But what really makes up most of that sequence is repeats, which completely makes sense for a non-recombining region to be riddled with repetitive elements. But kind of asking that question that we've been talking about this whole time of figuring out what are the sex determining genes. 3,400 genes is a lot to try and figure out which it is. So it's going to be a challenge. Maybe there's another species with potentially a smaller but homologous sex chromosome that we can leverage in the future. The cool thing, though, you can kind of see, so here's gene content along, excuse me, along each of the autosomes and the sex chromosomes. You can see TEs are elevated. When you kind of dig into some of these repetitive elements, some of the patterns we were finding were kind of interesting. You'd expect that some things, with an increased expansion in a region, that maybe all kinds of repeats would expand proportionally, but things like copia elements were pretty consistently, and they were non-significantly different between autosomes and sex chromosomes in the species. However, some elements were expanding in one sex or the other. So this Lanicia element is one that was identified in Ceratodon. This is a new TE. And it's very much enriched on the V chromosome, and these hat elements are enriched on the U chromosome. So it's really interesting potential follow-up for, is there something going on there that's there's a sex specificity for some TE expansions or any kind of cool studies like that, or is it coincidence? But because there's so many genes, it gives a really, really good data set in order to address how old are these sex chromosomes. So when you think about it, like I mentioned, mosses, 60% are dioecious. That's a lot of them. There's a lot of them kind of, peppered kind of across the phylogeny. These sex chromosomes are huge. It feels like there's the potential for them to be kind of old. And so what I did was I took a bunch of bryophyte data. So we took the annotations from Ceratodon, annotations from Fiscomatrella and Marcantia, because Marcantia is a liverwort, but also has a sex chromosome pair that's been identified. And then a ton of RNA-seq data, basically anything I could get my hands on that was a bryophyte, I used to build some gene trees. And just very briefly, what I did was assembled all of the RNA-seq data into genes, clustered orthologs using OrthoFinder, and built trees for every gene that was annotated on Ceratodon U and V, as well as on the Marcantia U and V. And so what we're going to look for is the patterns on these gene trees to identify how old they are. And so if something is only sex-linked in Ceratodon, we expect to see a pattern like this, where the Ceratodon male and females, they're non-recombining, so there are going to be differences that have been picked up between them. But you'll see that the males and females of Ceratodon are still sister to one another, and the rest of the species of the mosses here are following the typical topology. And so that means these genes became sex-linked after speciation from the sister taxa that I have available. And so they coalesce back here. If instead it's something that has an older origin, it showed up on the sex chromosomes prior to speciation, then we'll see a pattern like this, where you'll see something like Centrichia females more closely related to Ceratodon females than they are the other Centrichia isolates. And so you'll see this pattern of kind of a duplication, a mirror of the tree, where there's a U-linked copy, which will be the females, and then a V-linked copy in the males. And so that means became sex-linked before speciation, coalescence is further back in time. And so this is what I looked at thousands of gene trees, which are complicated to look at in and of themselves, but thousands of them was a lot, but found some very cool patterns. And so I'm going to orient you to how I'm going to show the results to show how old moss sex chromosomes are. I'm going to change the tree to just having a single representation per species. Here's Ceratodon, because that's the focal species we're looking through, which we know genes are sex-linked on. And so anything that I tick that's on this side closer to the names, that's going to be a more recent origin, and anything that's further back is going to be an older origin. And so when I looked across all of these trees, this is what I found. So most of the moss or Ceratodon UV genes were recent origins. So greater than 300 of them that I had really good and well-supported topologies to ascertain these results with. Those were Ceratodon specific based on the data available. But then there's multiple other capture events. So there's 13 that had evidence of being sex-linked before the dicronales evolved. And then a few more that go even further back. So these ones here that have pretty old origins. And found similar patterns with Mercantia, that there's multiple capture events. Some genes were Mercantia specific and some have a really far back origin. And so to put that in the context of like how old is that, use the Lehnen paper, so from 2014, where Lehnen et al. Were able to date using fossil calibrations all of the bryophyte phylogeny. And conservatively speaking, that furthest back node, the date that we're going with it's about 300 million years ago. So the evidence of those gene trees is that the sex chromosomes evolved about 300 million years ago it's before about 95% of moss diversity has evolved. Not every moss afterwards is dioecious, which is really interesting, and should be some fun follow-ups for if dioecy was like the ancestral state in this clade, what happened to the sex chromosomes. But these, at least when looking through Ceratodon's vantage, we find evidence that there's a really old origin, about 300 million years of these sex chromosomes. And so just to put that in context, that's when the Carboniferous area and Permian period was, so 300 million years ago. And if you look at this picture of these charismatic megafauna from back then, if you look real close at there's some tree branches on there, and I think there's moss on there. And if that's moss, it has sex chromosomes that have evolved. 300 million years ago, that's before the petrified forest. This is a national park down here. That's before it got scared. And that's still when Pangea existed. That's how long ago we're talking that the moss sex chromosomes evolved. And in the context of other species that have been studied, the bird sex chromosomes have evidence of being about 130 million years old. Mammals, the XY pair that is shared across mammals, 190 million years old. Flies and cockroaches are about 350, and I should add this because there's something that has beat us out, which is the liverworts. I showed you some of those trees, some of the results I had showed pretty old origins of liverworts too, but there's another recent paper that puts them at about 400 million years old, so probably the oldest pair that has been identified, if not the oldest pair at all. So it's up there with the old sex chromosome systems, which is really cool because most other plants up to this point, we'd found pretty recent origins, like Amberella was 16 million years. And so just to kind of wrap up all of the stuff that I presented today, we can now phase sex chromosomes and genome assemblies. We can phase them out with long-read data, scaffold them with Hi-C and Omni-C, and then be able to find the SDRs, the non-recombining regions, using k-mers. Amberella has a ZW system that evolved about 16 million years ago, and that non-recombining region we were able to find is about 3 megabases and has about 100 genes in it. The hop sex chromosomes are in progress, but at least as far as we can tell, based on comparisons with cannabis, is that it's a different XY pair than the Moraceae. So that'll help us narrow in on a potential date if it's not a shared origin, if they were truly independent. And then the Ceratodon, the mosses, have UV sex chromosomes that evolved about 300 million years ago. They're over 100 megabases long with 3,400 genes, and they are just chocked full of repeats. So very complicated, but very cool sex chromosome pair. And while these results are really, really promising, I really want to test this on more evolutions. And so that's kind of the reasoning behind this poster here is we want to sequence all the dioecious things. It's a big goal, but we want to tackle as many of them as we possibly can and find as many of those evolutions as we can and as many sex-determining genes. And it starts with being able to assemble out really, really nice phased sex chromosome pairs. So if you work on a dioecious species, I would love to talk about working on a genome assembly. We just got some funding to be able to fund some more genomes on top of the ones we're already working on. Please feel free to shoot me an email if you'd like to talk about dioecious genomes. And with that, I would like to again thank all of my amazing collaborators and funding sources, and thank you for your time.
Thank you very much, Sarah. It was very nice. I learned many new things about sex chromosomes. And of course, I'm more interested in bryophytes than all the others, but the others are fascinating as well. So are there any other questions?